I am Bhaskaran Pillai, founder of Agastiya Biotech, situated in San Diego. I have two other companies, OPEC and East West Research. OPEC is a pharmaceutical company, both allopathic as well as Ayurveda and Siddha, whereas East West is purely a research company. Both of them are in India. In this very short presentation, I want to address some ambitious goals as far as cancer therapeutics is concerned. Goal 1, damaging CSC, which is cancer stem cells, which is really a very ambitious goal because it's very difficult to find this cancer stem cells, and then to damage it is even a bigger problem. And because, as most people know, that cancer stem cells are always hiding, and then it is just only 1% of the cancer cells, and that's why we are not able to, first of all, find them. Secondly, to damage them is impossible. But the damage that it does in the disease progression is worse because it is responsible for relapse and renewal and drug resistance and so forth. And that is number one goal that I set myself to. The second one is toxicity. Everybody knows about toxicity of cancer itself. And also the toxicity of chemotherapy. People are afraid of chemotherapy more than cancer because the pain in the chemo treatment is unbearable. And that is the second goal. And the third goal is how to improve the immune system so that the cancer tumor will not be able to overpower the immune system. So if you make the immune system stronger than the tumor, then we have found a cure for cancer. So we have been able to accomplish in vitro as well as in vivo studies, not just on one cancer, but multiple cancers like pancreatic and lung, breast cancer, colorectal, AML, CML. It seems to be like I'm telling you a story, but it's true that we have scientific research to back us up. And then after my presentation, Dr. Dinesh, who is our chief scientist, will give you the research behind what I am talking about. And of course, we do need to prove it on the human model, which human trial we will be starting pretty soon, maybe as early as June. Now, the solution for all the problems came from just one molecule. And this one molecule also works for all these different cancers. And what is important is we tested on stage 4 level cancers of all these cancers, stage 4 metastasis. And then we have great results coming from that. And what is the secret behind this molecule, which I have named as AB001, Agastiya Biotech 001. It is the first molecule. It's not the only molecule that we have. We have several other molecules in the pipeline, and so which all seem to be very, very efficacious, and then with less toxicity, very negligible toxicity of all the molecules that we have discovered, or have negligible toxicity, meaning that it won't kill the normal cells. It will only damage the cancer cells and cancer stem cells. So why is this molecule special? This molecule is special because it combines uh, two different methodologies. One is the Siddha tradition. The Siddha tradition is at least 5,000 years old, discovered by the Siddha masters. Combines this Siddha tradition with Western biotech, resulting in a molecule that not only works on a molecular level but a particle level. So the future of medicine is going to be like a quantum medicine, where the molecules are intelligent and knows what it needs to do and goes to the target. You don't set the target, the molecule or the particle sets its own target, and that is new about this molecule. I hope that this medicine, which I call as the new molecule, will be the future medicine. Now I will introduce Dr. Dinesh, who is our chief scientist. His specialty is stem cells. It all worked out that we started with the stem cells, and so he was able to do a very good job with that. And here is Dr. Dinesh.
Thank you, Dr. Pillai, for giving this wonderful opportunity for carrying research work in Agastiya Biotech. Currently, we have been working with a novel small molecule inhibitor AB001, which has been identified for advanced stage metastasis cancer. Both in vitro and in vivo studies proved the safety and efficacy of the molecule in different tumor models compared with the standard anti-cancer drugs. Interestingly, this molecule selectively targets and eliminates the cancer stem cell population and exhibited significant tumor growth suppression in mice xenograft model. So, at the molecular level, this molecule downregulated the expression of various cancer signaling pathways, especially beta-catenin, Wnt signaling, Notch, and STAT pathway, which regulates stem cell self-renewal and proliferation. So here the slide 1, which shows the anti-cancer effect of the molecule AB001. So it showed a broad spectrum anti-tumor activity against a panel of various cancer cells, which was isolated from a variety of human cancer, including lung, colon, liver, breast, pancreatic cancer cells. Normally, this molecule as a most effective inhibitor of cell growth and proliferation with inhibitory concentration value for most cell lines was found to be in the range of 0.1 to 10 micromolar. So probably next we investigated the cellular and molecular events underlying the cytotoxic effect of this small molecule. This small molecule triggered the mitochondrial mediated pathway as indicated by changes in Bax BCL2 ratio, which resulted in the subsequent loss of mitochondrial membrane potential, cytochrome C release, DNA fragmentation, and caspase activation, which consequently caused cell cycle arrest, which was confirmed by flow cytometry, gene and protein expression analysis. Normally, the traditional therapy targets only the fast-growing tumor mass, but not slow-dividing cancer stem cell. It has been shown that the embryonic pathway such as Wnt, Hedgehog, and Notch control self-renewable capability and involved in cancer stem cell maintenance, and targeting these pathway may be effective in eradicating cancer stem cells and preventing chemotherapy and radiotherapy resistance. So the current study investigates the effect of AB001 with a focus on key stem cell markers involved in early cancer formation, stem cell maintenance, and differentiation. So for this study, the cancer stem cells were isolated and using flow cytometry and enriched by commercial kit according to manufacturer's instruction. The enriched cells were treated with AB001 for indicated times, and apoptotic response was measured. From the result, dose-dependent apoptotic response was observed in all the cancer stem cells treated with the small molecule inhibitor AB001. Normally, this reduction was observed within 2 hours of time, and no cancer stem cell markers was observed after 24 hours treatment, compared with the normally available standard drugs. So AB001 significantly attenuated the cancer stem cell tumor sphere formation, growth, relapse, invasion, and migration in vitro studies. To determine whether these small molecules abrogates the stemness of cancer stem cells, we examined the expression level of the cancer stem cell markers such as OCT4, NANOC, CD133, 44 and LGR5, which are the main regulator for cancer stem cell maintenance and renewal. So normally compared with the standard drugs, AB001 shows significant downregulation of stemness protein expression level in a dose-dependent manner, resulted in a significant reduction of the cancer stem cell population. To determine if the regulation of cancer stem cell gene is through the regulation of Wnt beta-catenin pathway, we determined the effect of beta-catenin and downstream effector gene TCF4 using real-time PCR analysis. Because Wnt beta-catenin signaling pathways plays critical role in the development of tissue homeostasis and self-renewal of stem cell and cancer stem cell genes. Our result revealed that AB001 inhibited Wnt beta-catenin and TC4 factors expression in a dose-dependent manner, suggesting that inhibition of marker expression by downregulating the expression of the Wnt target genes and finally leading to apoptosis. These results clearly suggest that AB001 target cancer stem cell population by regulating the expression of cancer stem cell gene at both the transcriptional and translational level. So, the relapse after chemotherapy treatment is a common phenomena. We hypothesized that the cancer stem cells are responsible for cancer relapse and that stemness inhibition can suppress cancer relapse. To effectively block cancer relapse, it is therefore essential to target cancer stemness. To evaluate the phenomenon of tumor relapse, we used a xenograft model and the chemotherapy agent gemcitabine. The treatment with gemcitabine inhibited the xenograft tumor growth with a tumor growth inhibition of 40.5% on day 45. However, after cessation of treatment on day 41, the tumors in the gemcitabine treated animals soon relapse and even outgrow the tumors in the vehicle control group. So treatment with AB001, that is our small molecule, also significantly inhibited the xenograft tumor growth even on day 21. However, unlike the gemcitabine treated animals, no tumor regrowth was observed in the animals administered AB001 during the 22nd post-treatment observation period. Furthermore, no signs of toxicity as evidenced by body weight measurements were observed. These data suggest that AB001, unlike other chemotherapy, can inhibit the cells within the tumor mass that are responsible for tumor relapse. So, AB001 showed an in vivo efficacy in various cancer models like triple negative breast cancer, acute myeloid leukemia model, pancreatic cancer model with a tumor growth inhibition of 90% without any mortality growth inhibition in comparison with other standard drugs. So AB001 prevent metastasis to the lungs and lymph nodes by inhibiting lymphoangiogenesis and angiogenesis in the pre-metastatic organs, whereas the standard drugs which shows limited access to cancer stem cells due to the hypoxia environment. Furthermore, this molecule inhibited the tumor growth of xenograft mice model and modulated the expression of mesenchymal and epithelial markers. It suppressed the expression of the mesenchymal genes such as fibronectin, vimentin, encadherin, TIST and snail, and increased the expression of epithelial genes such as ecadherin via specifically targeting Wnt beta 1, hedgehog, TGF and SMAD pathway. So next we did the pharmacokinetic studies with the AB001 which revealed the prolonged persistent in systemic blood circulation and no signs of nephrotoxicity, cardiotoxicity and hepatotoxicity was observed compared with other standard drugs which shows severe cardiotoxicity, hepatotoxicity, gastrointestinal toxicity and respiratory disorders. Based on the pharmacokinetics, the biodistribution and tolerability when compared with the standard drug administration. So in vivo biodistribution studies reveal that the accumulation of our small molecule in the tumor of the animal model was considerably higher than in the other organ analyzed. So next slide is related with the histopathological examination of the sections of liver, kidney, heart, brain and spleen from both sexes of acute and subacute toxicity groups were carried out on the final day of the experimental period. In this study, none of the organs showed morphological alterations or any other abnormalities in cellular architecture observed under the microscope. The ability of AB001 to kill both cancer stem cells and therapy-resistant cancer cells may define the compound as a novel and effective anti-cancer drug.